Have you ever wondered how the future of medicine and agriculture might look like? A future where we can expertly manipulate the building blocks of life to cure diseases and feed an ever-growing population. This isn't just a dream, but a real possibility with the help of gene editing technologies like CRISPR. Today, both medicine and agriculture face significant challenges. In medicine, we're constantly battling against diseases that mutate and resist our best treatments. In agriculture, we're tasked with feeding a booming global population, while dealing with climate change and dwindling resources. The stakes are high, and innovation is crucial. Enter CRISPR and other gene editing technologies. You might have heard of them. They've been making waves in the scientific community, and for good reason. These technologies have the potential to revolutionize how we approach these challenges. In medicine, gene editing could allow us to precisely edit our genetic material, potentially curing genetic diseases at their source. Imagine a world where inherited conditions like cystic fibrosis or Huntington's disease are things of the past. In agriculture, these technologies could help us create crops that are more resilient and productive, even in challenging conditions. We could engineer plants to better withstand pests or drought, or to yield more nutritious produce. This could be a game changer in ensuring global food security. But what is gene editing, and how does it work? At its core, it's a way to make precise changes to the DNA of a cell or an organism. It's like being able to find a specific sentence in a book, and then edit it to change the story. And the most popular tool in this editing process? That's CRISPR. So, how exactly does this gene editing work? Let's dive deeper. To understand the potential of gene editing, we first need to grasp what it is and how it works. So let's dive in. Picture DNA as an intricate instruction manual. Every organism, from a tiny bacterium to a towering oak tree, carries this manual within its cells. It's a blueprint that dictates every detail about the organism, from its eye color to its susceptibility to certain diseases. Gene editing, then, is like being able to rewrite parts of this manual. Imagine you could erase a sentence that leads to a genetic disorder or add a paragraph that gives a crop resistance to pests. That's the power of gene editing. Now let's meet our main character in this story, CRISPR. One of the most exciting tools in the gene editing toolbox. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Quite a mouthful, isn't it? But don't worry, it's not as complicated as it sounds. CRISPR is essentially a pair of molecular scissors. This precision tool can target specific parts of the DNA sequence, cut them out, and even replace them with new ones. The secret to its precision lies in two key components: the Cas9 enzyme, which acts as the scissors, and guide RNA, which directs the scissors to the right spot in the DNA sequence. Together they form a dynamic duo that can make precise edits to the genetic manual. The beauty of CRISPR is that it's not only precise but also efficient. It's like having a state-of-the-art word processor for our DNA manual, allowing us to make quick and accurate edits. Now that we know what CRISPR is, let's explore how it could change the world. Imagine a world where genetic diseases are a thing of the past. Sounds like a sci-fi movie, right? But with the advent of gene editing technologies like CRISPR, this could soon become a reality. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a revolutionary tool that allows scientists to edit genes with unprecedented precision and efficiency. In the realm of medicine, the potential applications of CRISPR are vast. It can be used in gene therapy, a form of treatment that involves altering the genes inside your body's cells to treat or stop disease. For instance, scientists could use CRISPR to remove mutated genes causing inherited diseases like cystic fibrosis or Huntington's disease, and replace them with healthy copies. CRISPR also plays a significant role in drug development. By better understanding our genetic makeup, we can develop more effective and personalized treatments. Drugs can be tailored to an individual's genetic profile, improving their efficacy and reducing side effects. It's like having a custom-made suit, but in this case, it's a custom-made treatment plan. And here's the real game changer: CRISPR could potentially cure genetic diseases. Yes, you heard that right. Diseases that have been deemed incurable because they're written into a person's DNA could be rewritten. We're talking about conditions like sickle cell anemia, muscular dystrophy, and even certain types of cancer. Of course, we're still in the early stages of this technology and there's a long road ahead. But the potential is there, and it's enormous. As we continue to unlock the secrets of our genetic code, the possibilities for healing and curing are expanding in ways we could only dream of a few years ago. The medical field is just the tip of the iceberg. CRISPR holds immense potential in agriculture as well. Feeding the growing global population is a daunting task, but CRISPR could be the game changer. Imagine a world where our food crops are resistant to pests and diseases, where the yield of our farms is significantly increased, and the nutritional content of our food is improved. This is not just a dream, it's a possibility with CRISPR technology in agriculture. CRISPR, a gene editing tool, is like a pair of molecular scissors. It's designed to precisely cut and edit the DNA of an organism. In the context of agriculture, this means we can manipulate the genetic makeup of our crops. We can engineer them to be more resilient, to withstand harsh climates, to resist pests or diseases, and even be more nutritious. Let's take an example. Say we have a strain of wheat that is particularly susceptible to a certain disease. With CRISPR, we can identify the specific piece of genetic code that makes the wheat vulnerable and then change it. The result? A strain of wheat that's no longer susceptible to that disease. But it's not just about disease resistance. CRISPR can also help to increase yield by making crops more robust and productive. It can even be used to improve the nutritional content of food. Imagine rice that has been genetically edited to contain higher levels of vitamin A, or corn with increased protein content. These are not just possibilities, they are realities being explored right now. Of course, like any revolutionary technology, CRISPR isn't without its challenges. There are concerns about the long-term impact of genetically edited crops on the environment and biodiversity. There's also the question of access. Who will have the right to use this technology, and who will benefit from it? The possibilities with CRISPR are vast and exciting, but they also come with their own set of challenges. Like any powerful tool, CRISPR also raises important questions and concerns. As we delve into the world of gene editing with CRISPR, we cannot overlook the ethical considerations and potential risks that come along with it. Gene editing technologies like CRISPR hold immense potential, but they also carry risks. One such risk is off-target effects, where the gene editing tool might unintentionally alter parts of the genome that weren't meant to be edited. This could lead to unexpected and potentially harmful outcomes. Additionally, we need to consider the long-term effects of gene editing. The changes made by these tools are permanent and could have unforeseen impacts on future generations. The ethical considerations are just as significant. For instance, should we permit gene editing to enhance human traits such as intelligence or physical appearance? This could potentially lead to a society where genetic enhancements become a commodity, accessible only to those who can afford it, widening the gap between the rich and the poor. Moreover, the use of gene editing technologies in agriculture raises questions about ecological balance. If we genetically modify crops or livestock, how will it affect biodiversity? Could it inadvertently harm other species or the environment? These concerns underscore the need for robust regulations and safeguards. Scientists, ethicists, and policymakers must work together to develop a framework that ensures gene editing technologies are used responsibly. We need to strike a balance between harnessing the potential of these technologies and minimizing their risks. Moreover, public engagement is crucial. Everyone should have a voice in the conversation about how and when these technologies should be used. After all, these decisions will shape the world we live in and pass on to future generations. The future of gene editing technologies like CRISPR is bright, promising a revolution in medicine and agriculture. But it's a journey we need to navigate with caution and responsibility. We're at the cusp of a new era, and with careful consideration and ethical practice, we can use these powerful tools to create a better future.